Hello aspirants. So today in this video we'll try to understand one of the most important topic in biotechnology related to agriculture. So we'll discuss about GM mustard. So GM mustard was in news because GEAC, that is Genetic Engineering Appraisal Committee, that is the regulator of biotechnology sector in India, had given approval for seed production prior to commercial release. Because of that, many articles had come with respect to GM mustard. So we need to understand that what is this GM mustard. See, with respect to application of biotechnology in agriculture, so production of GM crops is one of the most important application area. So in India at present we have only one legal GM crop which is grown. What is that legal GM crop? That is BT cotton. Only BT cotton is legal in India. No other crop, no other GM crop is legal in India, not even GM mustard. So GM mustard, if it gets the approval of GAC and if government gives the final approval, then it could become legal in India, but it is not legal even till now. But in all these GM crops, as an aspirant we need to understand that why are we going for modification, why are we modifying mustard, what is the purpose, and then we also need to know what is the technology, what is the method through which we are modifying GM or we are modifying mustard. So in this small video we will try to understand all of these dimensions. So we will try to understand this GM mustard. So before we understand GM mustard, let us just understand a brief context about mustard. So India annually imports around 55 to 60% of the vegetable oil that it needs, and that costs India around rupees 1,20,000 crore. That is a big amount for the exchequer. So India wants to reduce this amount by increasing the production of the domestic oil seeds. So mustard is one among the top 3 oil seeds in India. But even mustard, what is the problem that we are seeing? That the yield of mustard has remained stagnant over decades. So why the yield has become stagnant in case of India? There are some reasons. So one of the most important biological reason for stagnancy of yield of mustard is that it is a self-pollinating plant. Mustard is a self-pollinating plant. What do you mean by self-pollinating plant? It means that mustard mustard is self-pollinating. Self-pollinating, it means that its flowers has both the male part and the female part. So if this is the anther, this is the male part, the anther is the male part. This anther produces the pollen grains. So these are the pollen grains, and this is the female part of the flower. This is the female part of the flower. So this is your stigma. So when we say that mustard is a self-pollinating plant, it means that its flower has both the male part, this is the male part, and it also has the female part. So self-pollinating means that the pollen grain of this flower only fertilizes the ovum of this flower only. So what has happened because of this self-pollination is that the genetic diversity of the Indian variety has drastically reduced over the generations. So because of that, the yield has become stagnant. So this is one of the most important biological reason, that because it is a self-pollinating plant, the yield has stagnated. What are the other reasons? So, in the Indian soil there is deficiency of boron. Boron is a micronutrient which is needed by the mustard, but because of deficiency of boron, so the yield has also decreased. And also because the mustard growing areas are mainly the rain fed areas. Because they are rain fed areas, so they are mainly dependent upon monsoon, they do not have good supply of irrigated water. So, because of that the yield has become stagnant. So, what is the response to this? That what do we want? That we want that the yield should be increased. How we are trying to increase the yield? By genetic modification. In genetic modification, what are we trying to do? We are trying to create, or we are trying to make this mustard into, we are trying to convert it into a cross pollinating plant. So, from a self pollinating plant we want to make it a cross pollinating plant. Cross pollinating means that this female part could accept pollen from other plants also, from other mustard varieties also. So, what has been done? What has been done by Dr. Deepak Pental and his team? So, you must understand, or these are some factual things that you should remember also, that GM mustard has been developed by Dr. Deepak Pental and his team. So, Dr. Deepak Pental and his team, he was former vice chancellor of Delhi University. So, in a research wing of Delhi University, he and his team has developed this GM mustard. So, what has been done is that we have tried to make this self pollinating plant into a cross pollinating plant. So, what is the purpose of making it cross pollinating? The purpose is that we want to introduce some hybridity, that is, we want to cross it with some other varieties of mustard. So, what we have done? We have taken a European variety of mustard. So, we have taken, this is, let us say, your Indian variety, this is the Indian variety of mustard, and we have taken a European variety of mustard, and we want them to get crossed. So, just to mark it out, so this has the anther and this has the pollen. What we want that this Indian variety should be able to accept the pollens from this European variety. So, then it will become what? Cross pollinating. So, how we have achieved that? We have achieved that by adding one gene called as the Barnase gene. This gene does what? It makes this part sterile. It makes the male part of the Indian variety sterile. Since we are adding this gene, the male part becomes sterile, or the pollens that it produces, they are sterile. So, these pollen grains will not be able to fertilize the ovum of this plant. So, now the female part can accept, or it will accept, the pollen grains from the wheat from the European variety. Because it is now able to accept the pollen grains from the European variety, what we will get is eventually a hybrid. We will get a hybrid variety. We are getting hybrid variety, and this hybrid, scientifically we are calling it as what? DMH11, Dhara mustard hybrid 11, or GM mustard is also called as your Dhara mustard hybrid 11. Dhara mustard hybrid. Why it is called as hybrid? Because we are crossing it with the European variety. Now, once we get this hybrid, what is the problem that we will face? Because we have added this Barnase gene in this Indian variety, this Barnase gene will also be present here. This Barnase gene is present here. So, again what it will do is that it will make this hybrid variety cross pollinating, but we do not want that this hybrid should further cross pollinate. We just want that after we get the hybrid, we want that it should become self pollinating only. So, what we are doing is that we have added another gene called as Barster gene. Barster gene Barster gene will reduce the effect of the Barnase gene after the hybrid has been formed. So, this Barster gene will reduce the effect of the Barnase genes. So, it will make the male part fertile again. So, when the male part becomes fertile again, so self pollination is possible. So, the idea here is that we add Barnase gene to make it cross pollinating, then we are adding this Barster gene to again make it self pollinating after it has become hybrid, and we are adding one more gene that is called as the Bar gene. So, we have added 3 genes in GM mustard, and these 3 genes that we have added, we have taken it from a bacteria known as Bacillus amyloliquefaciens. So, we have taken it from bacteria Bacillus amyloliquefaciens. So, we have taken 3 genes from this bacteria, and we have added it to the hybrid. So, Bar, what is the role of Bar? It is making the mustard herbicide tolerant. It is making the mustard herbicide tolerant. We have seen the roles of Barnase and Barster. So, in you would often see that in GM mustard we are using Barnase Barster system. You would be seeing that we are using Barnase Barster system. This is a general technique to make any self pollinating plant cross pollinating. So, this technique we have used in GM mustard also. This Bar gene, what is the role of the Bar gene? Bar gene is making the mustard herbicide tolerant. What are herbicides? Herbicides are chemicals which are added or which are sprayed in the fields to kill the unwanted plants. Unwanted plants, that is the weeds. So, the objective is to remove those unwanted plants. So, why we are making it herbicide tolerant? We want to make it herbicide tolerant because we want that if we add herbicides, then it should kill the unwanted plants and not the mustard plant. And also farmers want this because in the normal crop cycle, a farmer has to spray twice or thrice during the crop cycle life this herbicide. So, this increases the input cost, this increases the labor cost. So, what farmers want, that they should be able to add more quantity of herbicide at one go itself, so that they do not have to spray periodically these herbicides. So, because to reduce the labor cost, farmers want that the mustard should become herbicide tolerant also. So, this Bar gene has been added for that purpose. So, we have added 3 genes from a bacteria Bacillus amyloliquefaciens. And what are the advantages that we have got? So, what we have seen is that the yield has increased. The yield of the mustard has increased to about 30%. And also the nutritional value was found, that nutritional value has also improved. And because it is herbicide tolerant, it will reduce the cost, labor or input cost for the farmers, and overall for the Indian economy, how it can be beneficial, because the yield is increasing, so it can reduce down our import bill. So, this is how this is proving to be, can prove to be beneficial. But there are many groups which are opposing introduction of GM mustard. So, there is a group called as Coalition for GM Free India. Coalition for GM Free India, what are they claiming? That GAC has given approval without doing proper studies on impact of GM mustard on environment and human health, specifically on how it would be impacting the pollinators, how it would be impacting honey bees. So, they haven't done out exhaustive studies, or no independent or third party studies have been done to know the impact of this on environment and human health. So, that is their criticism. One of the other criticisms that they are pointing out is that this matter, so GM mustard, so a case is pending at Supreme Court. So, Supreme Court hasn't given its decision with respect to GM mustard. So, this body, Coalition for GM Free India, is saying that if the matter is sub judice, then why GAC gave approval in between. So that is also one of the criticism. And the biggest criticism GM mustard has attracted is because of this herbicide tolerant. Why? Because of this there is a concern. See, herbicides are chemicals. Herbicides are harmful chemicals which kill the weeds. But these herbicides, most of these herbicides are carcinogenic in nature. Carcinogenic means they are cancer causing. Because they are carcinogenic, there is a concern that if we make it herbicide tolerant, then the farmers would get an incentive to use excess herbicides, because they would be using it once during the crop cycle, during the crop life. So they would be incentivized to use more of this, more of herbicide. So if more or excess of herbicide is used, then it could seep into the soil, or it could percolate into the soil, eventually entering into our water bodies and can even enter our food cycle. So it would damage human health, or it could cause cancer. So this is the biggest concern with respect to GM mustard. This is why there is a lot of opposition, and because of that government has not given approval or the final approval to release this GM mustard. See, one of the important thing that you should know, that cotton is legal, BT cotton is legal in India. GM mustard, what is the difference between cotton and mustard is that cotton is a non-food crop. Cotton is a non-food crop, but mustard, if it is given approval, it will be a GM food crop. So there is a whole another debate arising that whether we should allow genetic modification in food crops or not. And if you look at the parliamentary committees, there is a parliamentary committee on science and technology. It has come out with a report on impact of GM crop on environment, and it has said, it has pointed out that 17 out of the 20 most developed countries are not going for GM crops. So why India should be going ahead? But again, so we need to do more discussions and deliberations, then we should evaluate that whether GM crops would be beneficial or not. So this is with respect to GM mustard. So let us even look at some of the other dimensions. Through these slides we will just quickly see what we have already seen. So in this slide it is saying that GM mustard is among the 3 largest oilseed crops of India, soybean and groundnut being the other 2, but the yields has remained stagnant. It is a self-pollinating plant. It is difficult to pollinate from the male part of another parental line until one creates a female only by introducing this male sterility in a parental line. So this male sterility has been brought about by adding Barnase gene. Okay, so GM mustard is also called as Dhara Mustard Hybrid 11. It has been developed by Dr. Deepak Pental and his team, and this project was supported by National Dairy Development Board and Department of Biotechnology. So it will help farmers to increase mustard productivity, which has been stagnant. So 3 genes, you can just take a note of these 3 genes, Barnase and Baster, have been derived from Bacillus amyloliquefaciens. The male part of the flower of the one of the parental line is made sterile. So we have seen how it is made sterile by adding this Barnase gene. And what are the advantages? Advantages, productivity will increase. This we have seen. It will reduce dependence on importing of oil seeds. Fortification will increase the nutritional content also. So we can go for fortification. What is fortification? Fortification is addition of nutrients. So fortification generally is done by adding externally some of the nutrients, but in such type of fortification, these are called as biofortification. Biofortification, it means the plant itself adds some of the nutrients, but we are adding some genes and those genes are helping to add some nutrients. So these are known as biofortification. So biofortification can increase the nutritional value of the mustard also. Next, it will be state controlled, so it will be available to farmer at affordable cost. So what are we trying to say here? See, it will be state controlled. So when we are saying that it is state controlled, when we are saying that it is state controlled, see, with respect to cotton, with respect to BT cotton, BT cotton was developed by a group of companies. The leader was Monsanto. Monsanto has developed BT cotton, and they have developed what is called as terminator seeds or suicide seeds. Terminator seeds or suicide seeds. What does this mean? They are terminator seeds or suicide seeds. It means that the seeds can grow for once only, and when these seeds they flower, the flowers would produce seeds, but those seeds would be sterile. It means that the farmer cannot use those seeds to grow another cycle. So it means that the farmer will always have to buy seeds from this company. So with respect to cotton, we are facing this scenario. They have created terminator seeds or suicide seeds by deliberately adding some genes to it. So farmers are dependent on seeds for every crop cycle. Now what we are saying here, because it will be state controlled, because it has been developed by a government funded research organization, and because it will be state controlled, these seeds will not be what terminator type, these seeds will not be suicide types. So we can say that it will reduce the exploitation of the farmers also. So that is another advantage of the GM mustard. Another point that we are saying that this variety of mustard will be more herbicide resistant than the normal mustard. So this advantage we have already seen, that it will benefit the farmers, so it will reduce their labor cost or the input cost, and it would protect them from drudgery also. So in 1 year they have already asked this question. So let us just look at the question. I know the font size is little small, so I will read it out for you. So this question has already been asked on GM mustard. So let us try to solve this question. With reference to genetically modified mustard, GM mustard developed in India, consider the following statements. This has been asked in 2018. GM mustard has the genes of a soil bacterium that gives the plant the property of pest resistance. Gives the property of pest resistance to a wide variety of pests. See, when we talk about GM crops, the first GM crop that comes to our mind is obviously BT cotton, and in BT cotton we have added some genes to make it resistant to pests. So there is a perception that in genetically modified crops we are only doing, or we are only making the crops pest resistant, but that is not the case. Genetic modification can be done for different purposes. With respect to mustard, the purpose is to increase the yield. With respect to cotton, the purpose is to make pest resistant. With respect to, if you take potato, in potato also we have modified potato to increase the nutritional value. So we have added protein to potato, or we have added genes which will increase the protein content in potato. That is called as protato. In rice, you would have heard about golden rice. So in rice also we have added some genes which is helping rice to add beta carotene or provitamin A. So we are trying to increase the nutritional value. In another variety of rice, we are trying to add genes so that it can grow even in haline or saline environments also, in saline soils also. In some cases, in case of tomato in USA, there is a particular tomato called as the Flavor Saver tomato. It is a GM tomato. What is the objective? To increase the shelf life of tomato. So in all the GM crops, the purpose is not one, the purpose is varied. So in case of your mustard, the purpose is to increase the yield. So here this first statement is not correct. So the first statement is not correct, so you can eliminate two options. So you are left with option B and D, 2 only, and 2 and 3 only. So we will check for the second statement. So GM mustard has the genes that allows the plant cross pollination and hybridization. Cross pollination and hybridization. So yes, this is true. We want to make it cross pollinating so that it becomes hybrid. What is the advantage of hybrid? Hybrid means that it has genes, or it has increased the genetic diversity. Because of increase in genetic diversity, so the yield also increases. In case of cows also, you would have heard that we have taken Indian cow and we have taken foreign cow, and we have got this hybrid, and this hybrid is producing quite a lot of more milk than compared to Indian cows and compared to those European cows. So they are hardier also. So the genetic increase in genetic diversity is increasing the fitness level or increasing the desired traits or qualities. So that is the idea even for mustard also. Okay, so this second is correct. So now we just need to check for the third one. GM mustard has been developed jointly by IARI and Punjab Agricultural University. So it has not been developed by these institutes, it has been developed by Dr. Deepak Pental and his team working at Delhi University. So Delhi University would have been the correct answer. This is not the correct option. So the third statement is also incorrect. So the correct answer in this case is B only. That is your second only. So let us just also look at GEAC. One question previously had been asked on GEAC. So let us learn through that question only. Okay, we will come to this. Let us look at this question. So this question is saying Genetic Engineering Appraisal Committee is constituted under. So GEAC is also in news because when it gave approval for seed production, so GEAC always keeps coming in news. So you should have a basic idea of what is this GEAC. GEAC is the regulator of biotechnology sector in India. Is it a statutory body? Is it a statutory body? When you say statutory body, it means has it been made under a law? So yes, it is a statutory body. It has been made under the Environmental Protection Act, EPA, Environmental Protection Act of 1986. It has been made under Environmental Protection Act of 1986. So what it is saying that GEAC is a statutory body constituted under. So there is this famous rules. So the name of the rules is quite big, but you can just remember that there is this 1989 rules. So there is this 1989 rules, and obviously there is this Environmental Protection Act of 1986. So it is notified under EPA 1986 and it follows the rules of 1989. What is the full name of the rules? Rules of the manufacture, use, import, export and storage of hazardous microorganisms, genetically engineering organisms or cells, 1989. So yes, it is a statutory body which is notified under EPA and it follows this rule. It is a regulator, but the uniqueness about this regulator, it is not an independent regulator or it is not outside any ministry. If you take the capital markets, if you look at capital markets, so SEBI is a financial regulator of the capital market, but it is not under, let us say, Ministry of Finance, but this body is under Ministry of Environment, Forest and Climate Change. So it is working under a ministry. This is also a noteworthy point that it is working under a ministry only. What are the different roles of this, or what are the functions of GEAC? The prime function of GEAC is what? Giving approval for field trials. So when there would be technology developers. Technology developers means there could be companies or there could be research organizations. Research organizations could be private owned or they could be government funded. So they are technology developers. They will develop a GM crop. Then they will go to GEAC. They will go to GEAC and ask permission for field trials. So then GEAC would give permission for field trials. After the field trials, these technology developers would provide data to GEAC. Then GEAC will study those data. It will study those data and try to find out the possible impacts of this particular GM crop on human and environmental health. And after that study, it can give the final approval whether this GM crop has to be released commercially or not. But the final, you must also take a note that the final approval is eventually given by Government of India. So in case of, if you would have remembered that in case of BT brinjal, GEAC was in favor. GEAC had given approval also for commercial release, but because there was wide protest in 2007, so BT brinjal was not released eventually by government, or it was not made legal by government. So yes, GEAC is the final approver, but the final authority for releasing of GM crops eventually lies with the government. So GEAC also has punitive powers. It also has punitive powers. It means that it can punish agencies or individual farmers also which violates the norms. So it has punitive powers also. But there are some criticisms also with respect to GEAC. Or what are the criticisms? So, recently there was a news that pointed out that this in rice, in case of rice, oh yes, this rice, GM rice, so there was a containment, or a container was sent to Europe, and in that container it was found that it had come from India, but it was containing GM rice, but GM rice is not legal in India. So GEAC it attracted some criticism, and it was said that GEAC has not been able to do or carry out its function. So because of that, GEAC was also in news, and previously it was also found that HT-BT cotton. HT-BT cotton is another variety of BT cotton in which we have added some genes to make it herbicide tolerant. Again, why we are making it herbicide tolerant? It is because we want that the farmer should be able to spray the herbicide at one go itself. But this is not legal in India, and again in that case also GEAC had attracted some criticism. So because of that, so there are some criticisms, so there are some suggestions also. So there is a Parliamentary Committee on Science and Technology which gave a report on GM. So it has said that there needs to be some reforms in GEAC. So reforms such as the chairman of GEAC should be an expert in this field. At least he should be an expert in biotechnology, and the selection procedure should be transparent, and all stakeholders like state governments, other NGOs should also get a say in the selection committee. So these are some of the suggestions, and it also points out that the data which GEAC gets from the technology developers, sometimes there could be a case of tampering of the data. So the field trials should be conducted under the supervision of GEAC, so that the data that they are receiving is authenticated data. So this is in short with respect to the GM mustard. So thank you.